Hello friends, welcome to another lecture series of translation in prokaryotes or the protein synthesis in prokaryotes. Today we'll be studying about the tRNA charging in prokaryotes. In the last video lecture we have already studied about the overview of the translation in prokaryotes, and there we have known what is mRNA, what is tRNA, and the different components involved in protein synthesis. So first of all, in this lecture series we'll go with the initiation part where we'll study about the tRNA charging and formation of initiation complex. And for our own convenience we have divided the initiation into two parts, the tRNA charging and the initiation complex, and today we'll only study about the tRNA charging. The next video will go with the initiation complex, and finally later on we'll go with the elongation and finally with the termination. So now what is meant by tRNA charging? Before knowing the process of tRNA charging, the steps of tRNA charging, we must know what is tRNA and what is tRNA charging. So the main components involved during the process of translation is, one of the main component is tRNA, which is also known as the transfer RNA or the carrier of the amino acids. Okay. So when you see the structure of tRNA, it is like a T shape or like a X shape molecule where we have different loops, and also there are different ends. One is known as the 5' end and the other is the 3' end. It will go from 5' end and will end towards 3' end. And in the 3' end we will have a acceptor site where we will have some sequences known as CCA. And in the left hand side, if you consider here, we have a D loop, and the downwards we have the anticodon loop, and in the right hand side we will have the T loop. So when you see the anticodon loops bear the anticodon site where they will attach to the codon complementary to it. Suppose if the codon bears UAC, as we know that the codon is formed from 3 nucleotide sequences. Okay, one codon is equal to 3 nucleotide sequence, and this one codon will bear one amino acid. So complementary to it will have, you will bear A, you will bind with A will bind with U, and C will bind with G. And so in the anticodon side of the tRNA here we will have AUG sequences. Okay. So now when you go towards the end part, that is in the 3' end, we will see a amino acid attached here. But this amino acid cannot be attached directly. It needs some mechanism before attaching in the tRNA. And this amino acid and this addition of amino acid to tRNA and the mechanism involved in it, in addition of this amino acid in a tRNA is known as tRNA charging. Okay. As you know that there are 20 different amino acids which will form the protein structure, or after the completion of the translation we will have the protein. So with 20 different amino acids we need 20 different tRNA. We also need to bring 20 different tRNA which will come one after another, and they will bring 20 different amino acids, and this amino acid will get attached with another amino acid and forms a peptide bond like structure, a polypeptide structure. And this polypeptide, after formation of 20 different amino acid, they will form a protein. So the steps of tRNA occurs in two ways. One is known as the adenylation of the amino acid, and the other is known as a transfer of this adenylated amino acid towards the tRNA. Now we'll know how the adenylation process is done, or the addition of adenine group to the amino acid. See, the amino acid cannot be attached directly to the tRNA. That needs to be, they cannot do it from their own self. They need to be activated, and this activation is done with the help of ATP, which bears the adenine group, and they will put this adenine group to the amino acid, and thus they will form the adenylation. They will form the adenylation of the amino acid, and this adenylation of the amino acid will activate the amino acid, and they can now go and bind with the acceptor site of this tRNA, okay, of this tRNA. So now when the ATP interacts with this amino acid, they will add the adenine group to the amino acid, and they will release PPI, okay, pyrophosphate, from the ATP after the reaction is completed. So this is the first step that is done during the tRNA charging, okay. In the next step, we see that this adenine group added to the amino acid should be transferred to the tRNA, and this process known as adenylation, okay, they need to be transferred to the tRNA, because without addition of this amino acid to the tRNA cannot go and bind with the, if they bind also to the anticodon loop, they cannot form the polypeptide bond, they cannot form the amino acid, and they cannot form the protein synthesis, okay. So how the adenylated amino acid is added to the tRNA, okay. Before knowing the mechanism, we should know what is the structure of amino acid. You see here, the amino acid consists of a R group, which varies according to the different amino acids. It also contains the NH2 group and carboxylic group, also a hydrogen group, and in between them they have a carbon group. These amino acids that gets activated by the addition of this ATP molecule, or the addition of adenine, will go and bind with the hydroxyl group of the 3' end of this tRNA, which consists of a CCA sequence, okay, and they will bind with the hydroxyl group, and they will bind with the help of ester bond formation. This ester bond is formed between the hydroxyl group of the 3' of the CCA of the tRNA and the alpha carboxylic group of the amino acid. So this way they will form a ester bond, and they can now bind with the tRNA, and this is catalyzed by an enzyme known as aminoacyl tRNA synthetase. Aminoacyl tRNA synthetase. Here, sorry, I have written it mistakenly. It will have E here, synthetase, not synthase but synthetase. This aminoacyl tRNA synthetase helps to catalyze or helps to bind the amino acid with the tRNA, and thus they can they can bind with the tRNA, and thus they can start with the initiation complex. They can begin the initiation complex formation, and finally they can go with the elongation process and do the translation and do the process of translation. So this is how the process of tRNA charging is done in case of prokaryotes. So I hope this video lectures is understood by you, and in the next video I will come with the initiation complex formation and how the initiation takes place, and finally we'll go with the elongation and termination of translation in prokaryotes. So please, if you like my video, do share with your friends and also subscribe to my channel. Thank you.